Okay.
Excellent. Thanks, Sam, for the great introduction, and thank you all for taking the time to join us today. As Sam mentioned, I'll be focusing on preclinical cancer research and the Prospect T1, the high-frequency ultrasound system, and taking a look at different tumor models. So again, we'll spend the first little bit focusing on different tumor models and how people are doing cancer research currently. We'll focus a little bit of time on imaging and preclinical research, highlighting many different imaging modalities, but then focusing very much in on the high-frequency ultrasound system. And then at the end, I'll take some time to look at some of the recent publications using that system in preclinical cancer research. So to get started, let's take a look at different solid tumor models. And I'm going to focus more on solid tumor models rather than leukemias and different soft tumors, for example, blood-borne tumors, just because those are the ones that we typically do image with high-frequency ultrasound. So first, I'll start and look at cell line-derived tumor models, moving on to genetically engineered mouse tumor models, PDXs or patient-derived xenograft tumor models, and finishing with a little bit of time on metastatic tumor models as well. So really the goal of preclinical research, why we're all doing that research, is really to identify the biological pathways and the different mechanisms that are important to tumor genesis, growth, and metastases. Alternatively, groups may be looking at screening for therapeutic compounds, but the main goal of preclinical research, preclinical cancer research, is to take what's found and translate it into the clinic to try to bring new therapies for those patients who happen to develop cancer. So there was a paper published in 2017, and the group looked at a number of publications from 2016, and they made some summaries and some conclusions on how cancer research was being performed. So looking mostly at solid tumor models, they were able to see that a very high percentage of the papers used cell line derived models, in fact 82%. So it's very high in terms of that model being used to do a great deal of the research in the community. Patient-derived xenografts are starting to pick up pace in terms of people using them, and we'll talk about perhaps why that's the case. But again, this was only 7% of the papers used PDX models. Moving on to environmentally induced models, a slightly lower percentage at 6%. And finally, genetic engineered models bounced back up to about 24%. And we'll go into each one of those models in a little bit more detail. Then they took a look at the types of tumors that were being looked at and the models that were being used. And there was a clear indication that most people were just either looking at a single tumor model or a single tumor type. The most common type of tumor that was imaged was breast, followed by lung, colorectal cancer, melanoma, pancreas, brain, and prostate. And then what they did was they took these types of tumors and they looked at what model was used to investigate them. And we can see across the board the most common was, again, the cell line derived tumor models, with other PDX, genetically engineered, environmentally induced models being used at different percentages. The clear difference there is the pancreatic tumor model. There's a really great genetically engineered model that mimics the human disease quite closely. And so that model is used quite extensively for pancreatic tumor imaging. So let's take a look at each one of these types of models very briefly as we go through. So cell line derived tumor models are really the workhorse, as we saw throughout those slides, and as most of you understand from the work that you are doing in preclinical cancer research. And the reason for that is that they're propagated easily. They grow well in vitro, but they also grow well in vivo when they're implanted in immune deficient animals. The cells can be injected subcutaneously to form a tumor, orthotopically, and that becomes important when you're looking at the microenvironment that the tumor cells are growing in. Or they could be injected IV to cause a metastatic tumor model. Many of these cell lines can be and are genetically engineered to contain an enzyme, for example, luciferase, to allow them to be imaged by other imaging modalities such as bioluminescence, which is not possible, as we'll talk about, with some of the other tumor models. However, the downfall of the cell line derived tumor models is that they may not correlate well with the clinical outcomes for all patients. And that's really because the cell lines are a single cell type. And so the tumors are homogeneous and they only represent a single form of cancer, where we know that within the clinical population, tumors have such heterogeneous cells that make them up, as well as each cancer patient has a unique reason for the cancer being formed. So moving on to genetically engineered mice. So here there's two different ways that engineering is typically used. So either an immune deficient mouse has been repopulated with a humanized immune system, and that allows researchers to study specific parts of the immune system that may be important in tumorigenesis or propagation or metastases. Alternatively, the mouse may have a genetic mutation that allows for either tissue-specific or temporal control of a specific oncogene or loss of a tumor suppressor gene. There's lots of different manipulations that are done there. And this allows for the spontaneous formation of tumors, and they more closely mimic the human disease. The downfall here is that those tumors are variable in their formation, and they can take a lot longer. So you don't actually know when that tumor is going to form. Ideally, though, this allows the researcher to study all of the different stages of tumorigenesis. So looking at the interaction between neoplastic cells, as well as the environment that they're located in, all the way up to when a full solid tumor does form. Moving now to look briefly at patient-derived tumor models, or PDX tumor models. These are formed when a tumor is resected from a patient, and that tumor is broken up into small parts, and it's implanted into an immune-deficient mouse. And in this way, the tumors begin to grow within the mouse, and they can actually be expanded and grown within the immune-deficient mice through different passages. This is really the only tumor model system that allows for researchers to study the vast differences between tumors within the clinical population. So both the difference in cells within the tumors themselves, as well as the different types of cancers that may be found within the patient population. What's really unique about this model is that it can be used to test the efficacy of different therapeutic compounds. So if a patient was diagnosed with cancer, they may have part of that tumor resected or all of it, but then be enrolled into either a specific trial or put on a specific therapeutic compound, for example. But then in parallel, both in vivo and in vitro, those little portions of the tumor that were resected and divided up can be used and screened for other therapeutic compounds, which may be more effective than the treatment that that patient's on. So again, the cells could be studied in vitro for response to a specific chemotherapy, or the avatar mouse, which is a PDX tumor model, could be treated with different therapies, perhaps some trial compounds, to be able to see if that tumor is responsive. And then ultimately the patient could be moved on to that therapy to help cure their cancer. Looking a little bit closer at metastatic tumors. So this group wanted to see, when metastases was studied, where was the primary tumor placed? Was it placed orthotopically, subcutaneously, or ectopically, or both within the paper? But even more interesting was that only about 25% of those papers actually looked at metastases at all. So whether it was that they were studying the primary tumor and happened to look at metastases, or the primary role of that paper was to look at metastases. As I alluded to before, metastases can be induced in several different ways. One of them is by injection of a cell line, or a cell-derived tumor line. And this can be done either by an IV injection through the tail vein, intracardiac injection, so a direct injection into the blood pool, injection through the splenic vein, or just an IP injection into the peritoneum. And then at that point, you would look in specific target organs for tumor development over the course of days or weeks following. Alternatively, you can have a primary tumor that may be placed in the animal. Again, it could be subcutaneous or orthotopically placed, and then look for metastases to form in other organs. The issue here is often that the primary tumor may grow so quickly that it may overcome the animal. And so to study the metastases, these primary tumors often need to be resected to allow that animal to survive long enough for the metastases to take hold. I'd like to take a quick break now prior to moving to talking about imaging in preclinical cancer research. And I'll turn things back to Sam.
Thank you, Tonya. We're going to launch a quick poll. It's a two-part poll. First question is, what types of tumor models are you working with? And there's a couple choices: cell line derived tumor model, patient-derived xenograft, environmentally induced models, genetically engineered mouse models, or other? The second question is, what type of cancers are you focusing on? And then we have a bunch of choices there: breast, lung, colorectal, melanoma, pancreas, brain, prostate, liver, metastasis, or other. We're going to give you just a couple more seconds to allow others to answer the poll question. There's a lot of different choices being selected. Just a few more seconds. Okie dokie. Back to you, Tonya.
Great, thanks, Sam, and thank you both all for taking the time to answer those questions for us. It really does help us understand the audience that we're presenting to. So now I'd like to shift gears a little bit and take a look at imaging in preclinical cancer research. So we'll start by looking at all of the different imaging modalities that are typically used, and then I'll spend a lot of time focusing in on high-frequency ultrasound, specifically looking at the Prospect T1 and the capabilities of that system. This is a high-frequency ultrasound system that can be used to detect tumors, monitor their growth, look at the surrounding tissues, and I'll show you some examples of all of that. As well, we can do image-guided injections, and these can be used either, as I was talking about with the metastatic tumor line, to do intracardiac injections, or you can directly inject into the organ of interest to do an orthotopic placement of your tumor model. We'll then take a look at different ways to measure tumor vasculature and biomarkers within those tumor cells. As well, we'll take a look at some advanced techniques for drug delivery and early lesion detection. So focusing first on imaging as it's used in preclinical research, specifically within cancer research. So micro-CT. CT is X-ray based, and it provides really great resolution on bones and lungs. However, there's a great deal of radiation exposure to the imaging subject, which may or may not affect the study that you're conducting. PET imaging uses radioactive tracers, so there is still some radiation involved. And additionally, it simply provides a signal, and it should be co-registered with something that provides some anatomical context so that we understand where that signal was coming from. However, this is a highly sensitive technique that's used clinically as well as preclinically to detect metastases, as well as some early pathological changes that may be occurring within the tumor model. Bioluminescence, as I alluded to before, requires the expression of a specific enzyme, in this case, luciferase. And in this way, we can only use cell line derived tumor models because we have to affect the genetic expression of the luciferase. In this case, we're looking at the light intensity and how big the tumor is and associating the two. And so it's really important when using bioluminescence imaging that we understand the expression of the luciferase enzyme as well as the delivery of the luciferin to those tumor cells. And in some situations, as the disease progresses or responds to therapy, those may change, and that needs to be considered and taken into account when doing this type of imaging. When we take a look at MRI, this provides excellent soft tissue resolution and allows us to visualize both the normal and the abnormal structures, including, of course, the tumors themselves. Acquisition times vary from between 2 to 5 minutes or longer, and we get fairly good resolution, about 150 to 300 microns, covering the entire body. Contrast agents can be used to study the perfusion within tumors as well as specific biomarkers. And finally, we'll talk mostly about ultrasound today. And so specifically, high-frequency ultrasound provides high-resolution images, and there's a strong soft tissue contrast, allowing us to visualize the tumors compared to the normal tissue. Contrast agents, as you'll be able to see from many of the publications that I'll talk about later, allow you to look at both tumor perfusion as well as the expression of specific targets. I will make a side note here in terms of Scintica's offerings. Scintica actually has options for all of these different imaging modalities, but today, again, we're going to focus mostly on the Prospect T1 system, which is our high-frequency ultrasound system. So for those of you less familiar with ultrasound and how it works, ultrasound is a non-invasive imaging technique, and it doesn't require any radiation. Instead, it uses sound waves. So the ultrasound probe, very similar to a clinical system, is placed on the skin of the animal using an ultrasound coupling gel, and that just simply allows the sound to be sent and received. And that's exactly what the transducer does. It sends out sound, it receives the sound waves back, and then the computer interprets that into an image for us. Why high frequency, and why do we need to use high frequency for small animal imaging? It really comes down to the size of the target that we're looking at. Often the tumors that we're looking at or the structures within the tumors that we're looking at are quite small. And so high frequency ultrasound provides us with high resolution images. The trade-off always with ultrasound is that as you increase the frequency of sound, you do increase resolution, but you decrease penetration. And so that's where we don't use the same frequencies on humans as we do on mice and rats. With the frequencies used on the Prospect T1 and other high-frequency ultrasound systems, it's really no problem to see any of the anatomical structures within both mice and rats. So focusing now more on the Prospect T1. So again, this is a high-frequency ultrasound system designed specifically for small animal imaging. There's 3 different probes available at different frequencies, giving us a resolution up to 30 microns. This allows us to visualize very small tumors and looking at early pathological changes. The system allows us to do B-mode imaging or brightness mode imaging, and that's simply a 2-dimensional grayscale image that allows us to visualize normal and abnormal tissues and differentiate the two. We can add a 3D motor, and that allows us to do multiple 2-dimensional images, which we then stack back into a 3-dimensional volume, allowing us to do tumor volume quantification over the course of a longitudinal study. I'll show you in a few slides a mount that can go onto the imaging probe that allows us to do image-guided injections, again, either for intracardiac injection for a metastatic tumor line or orthotopic implantation of cells within a specific organ. We can use both power Doppler and contrast agents to look at tumor vasculature. As well, we can use contrast agents to look at the expression of specific biomarkers, and I'll show you many examples of that in the papers that I look at towards the end of the presentation. And finally, we can integrate a sonoporation probe, and that can help with increasing cell membrane permeability, as well as to help with drug delivery, and we'll talk a little bit further about that as well. And shear wave elastography. So this is an advanced ultrasound technique that allows for the detection of early lesions, and I'll show you one example of that as well. So first, taking a look at B-mode imaging or brightness mode imaging. Again, this is a 2-dimensional grayscale picture that allows us to visualize the tumors as well as the surrounding normal structures. All types of tumors can be imaged. Here we see a subcutaneous tumor sitting underneath the surface of the skin. Here we're looking at an orthotopically implanted liver tumor. We can see the tumor being different in echogenicity or brightness on the image compared to the surrounding liver tissue, which has a very characteristic appearance. Within the 2D B-mode images, we're able to do 2-dimensional area and linear measurements to take a look very quickly at the tumor shape and size. We can also use 2D B-mode imaging to look at other structures. So we may want to, and it's very important, to understand what normal structures look like. So this is a normal liver. The tissue has a very characteristic appearance, and then there's little vessels, these black holes, and lines that you're seeing running throughout the liver. So that's a very normal looking liver. We can begin to identify abnormal structures within these organs. For example, this is the ovary, and these black holes that we're seeing are actually cysts. And I know that because water does not return a signal on ultrasound, so it looks very dark. Where soft tissue and different structures return the sound at different amounts, and that's where we get the difference in the grayscale intensity. It may also be important to not only image your tumors in the normal organs, but to look at surrounding tissues that may indicate more of a systemic change, and that may be seen both in a lymph node, for example, or within the spleen, both are which clearly visible. Both of these are normal, and you'll be able to begin to recognize abnormal quite quickly. Moving on from 2-dimensional B-mode imaging, we can attach this 3D motor to the animal handling stage. And so here we actually have the animal handling platform, which is heated. It monitors ECG and respiration, and the probe is held within the holder here, which provides full flexibility for positioning the probe. When we mount the 3D motor on the side here, we're able to basically translate the animal underneath the probe, acquiring multiple 2-dimensional images to stack back into a 3-dimensional volume. And from there, we can do some 3D volume calculations. So let's take a look at what that looks like. Here I'm sweeping through all of the different slices that were taken on this specific tumor model. And so here we're just sweeping through them. We can actually see the kidney sitting underneath. And what I'm going to do now is come back to the central plane where we have the largest tumor area. I'm playing here with some of the settings that can be used to help the 3D volume edge detection work better. And as I'm placing my points around, I'm doing a simple area on this specific slice. I'm left-clicking as I go around, right-clicking to finish. If for some reason I misplaced one of the points, I can simply pick it up and move it. I'm then going to switch to the perpendicular plane and do the same thing again. And in doing that, I'm helping to guide the software in terms of its edge detection and giving it a starting point. And then from there, you'll see that I'll hit the play button, and that will allow the computer to perform the reconstruction of the 3D volume. So again, I can adjust the radius and the alpha. Those are just simple parameters that help with the 3D volume contour. And then the system generates a 3D volume. We see that as a purple overlay on my cube, and also in the top you can see the 3D volume, which is measured. And from here, we're able to move this cube in any orientation, move all of the 3 different slices around to see different structures within the tumor, and of course, use that tumor volume to measure, to monitor the change in tumor volume over time within your study. Moving on to image-guided injections. So there's a mount which can be placed onto the imaging probe itself. And basically what that does is it allows any needle with a steel syringe to be mounted, or pulled glass capillary needle if you want it to be placed, and then you would manually advance it into the target of interest. As we can see on the image here, there's a steel needle going into the heart of a mouse. In this case, the user was actually trying to inject stem cells into the myocardium rather than advancing the needle entirely into the left ventricular cavity to do, for example, a metastatic tumor cell line injection. Additionally, you could, within an abdominal organ, for example, the liver, the kidney, pancreas, all of those types of organs, you could use the same technique to do implantation of your tumor cell line rather than opening up the animal and performing surgery to do that type of cell line placement. Moving on to looking at the tumor vasculature. So here we have power Doppler, and this is a technique that looks at the reflection of the sound coming from the red blood cells. And so you almost see a twinkling on the screen as these red blood cells are flowing through some of the larger vasculature. And power Doppler is able to pick that up and overlay that image on top of your traditional B mode image. So we're able to get an idea of the tumor vasculature. If we want to look at the microvasculature, we would need to use contrast agents. And so these are typically microbubbles, so they often have a lipid shell or a protein shell, and they've got gas on the inside. Typically, they're 2 to 3 microns in size. They stay within the vasculature of the system. Because of their size, they don't leak out of the leaky tumor vasculature. They do often and most frequently remain within the vascular system. They flow freely when they are untargeted, and we're able to look at the tumor vasculature. On the Prospect T1, there's 2 ways to image these contrast agents: reference subtraction, which I'll show you first, and subharmonic imaging, which I'll show you on the next slide. So in reference subtraction imaging, we take a baseline image where there's no contrast agent being injected, and we subtract it from the image where the contrast agent is being injected. And on this image, we're putting the color overlay within our region of interest, but on the outside, you can see the signal enhancement, very bright signal as the contrast agent's being injected. And then what we can do is draw multiple regions of interest. And so in this way, we can put a region of interest on normal tissue within the tumor itself, or a specific region within that tumor that we may want to see what that change in tumor vasculature is within that region. From there, we generate time versus intensity curves, so time on our x-axis, intensity on our y-axis. And in looking at when the contrast comes in as we move off of baseline, that can show us a delayed perfusion into that specific region of interest. The intensity tells us how much is there. So again, if that signal intensity is higher, then it's more highly perfused than an area that would have a lower signal intensity, for example. So moving now to subharmonic imaging. So this is another way to look at contrast agents. And this comes from the properties of the agents themselves. Being bubbles, when we hit them with sound, they begin to oscillate. And as they oscillate, they send back both the frequency of sound that we hit them with, as well as harmonics above that. So if we send in 20 megahertz, which we would do, we would listen for 40 megahertz. And what that does is basically removes the signal from the tissue, because the tissue does not send back harmonics. It's really the microbubbles that do. So it increases our sensitivity and specificity to looking at the contrast agents themselves. And so in this image, we have contrast agent injected. The tissue signal is minimal, if anything at all. And then when we draw our regions of interest, we know that our time versus intensity curves are coming strictly from the microbubbles which have been injected. So again, that shift to the right shows a delayed enhancement in that specific area, and a decrease in the intensity shows us a lower level of perfusion within that area of the tumor. There are ways to use microbubbles to do targeted imaging, and we'll cover those in the different papers that we review towards the end of the presentation. Focusing again back on microbubbles. So we talked about the microbubbles being gas-filled, and when they're hit with sound, they begin to oscillate and send back the harmonic sound. We can use microbubbles to cause what's called sonoporation. And this is basically the controlled cavitation or bursting of the microbubbles. And so when we hit them with a specific power of sound or energy of sound, the microbubbles don't just oscillate, they begin to burst. And we want to do that in a controlled way so that we're not causing significant tissue damage. But what it does cause is an increase in cell membrane permeability, as well as opening the blood-brain barrier. Additionally, if those microbubbles have drug contained inside them or within their shell, it can help facilitate drug delivery. And obviously opening the blood-brain barrier and opening the cell membrane also helps with getting agents across those barriers. So the Sonoporation probe is a secondary probe. It's a non-imaging probe, which is angled towards the tissue of interest. And then that probe is integrated within the software of the Prospect T1. And from there, we can control when it fires, how much energy it fires with, and many of the different settings which would be desirable to control the sonoporation event. An additional advanced technique is called shear wave elastography. And when we're doing elastography, we're looking at the changes in the mechanical properties or the elastic properties of a specific tissue. How this works is that there's an additional probe mounted onto the side of the imaging probe, and this is called the push probe. And what it does is it sends in a push pulse, an ultrasound pulse, and then it detects how fast that pulse propagates throughout the tissue. And so as the sound begins to propagate through the tissue, if it's healthy and normal, it's going to have a very specific speed. If there's a lesion or a change in how that tissue allows the sound wave to propagate through, then we're going to see a change in how fast the sound wave begins to propagate. And so we can see that in a colored elastogram, which is overlaid on top of the B-mode image. And so we see here the scale, so blue, green, yellow, red, shows us the change in speed of that propagation of that push pulse. And so the liver has a very characteristic appearance. If there's a lesion beginning to form, it's going to change. And this may allow for earlier detection of changes in the tissue, which may not be immediately visualized by ultrasound, but it indicates that something is going on and it should be investigated further and perhaps monitored over time. I'd like to turn things back over to Sam now for our second poll, just to learn a little bit more about the work that you guys are doing within your labs.
Thank you, Tonya. Thank you for that wonderful information. Our second poll is another two-part poll. First one is, are you currently using imaging in your cancer research? And the choices are yes, we're happy with our imaging capabilities. Yes, we're using imaging, but see room for improvement or additional imaging modalities. No, but we would like to add imaging to our lab. No, I'm just here to learn more about what others are doing. The second part is, what imaging modalities are you using in your research? And there's multiple choices. And the ones we talked about, CT, PET, MRI, bioluminescence, fluorescence, ultrasound, other, or none. Have a little bit over half voted so far. We'll just give you 10 more seconds to get the other ones to answer the poll questions. And it looks like we have mostly everyone, so I'm just going to bring Tonya back. Tonya?
Great. Thanks, Sam, so much. And thank you again all for providing us with a little bit more information about the imaging that you're doing, if you are doing any, and the models that you're working with. So now moving on to some of the publications that have more recently been done using the Prospect T1. So this group took a look at developing a theranostic agent. And so a theranostic agent is both a therapeutic and a diagnostic agent. And so really what their goal was to develop an agent which would accumulate within a tumor, allowing them to diagnose the tumor and visualize it more clearly. As well, they wanted to increase the retention of the contrast agent in the tumor, and that was to affect drug delivery. So be able to effectively deliver drugs specifically within the tumor, see a change in tumor growth, but perhaps not affect the systemic release of that agent across the whole animal, so affecting their well-being. And so what this group did was they made a contrast agent which had aspects incorporated from 3 different imaging modalities. So they included a marker for fluorescence imaging in the shell. They conjugated gadolinium, which is an MRI contrast agent, to the chemotherapeutic or the therapeutic agent that they associated with the microbubble. As well, that microbubble had a gas core, and so that allows it to be visualized under ultrasound. What they also did was to cause increased retention of the microbubble to the tumor itself was they put folate on the outside of the cell of the microbubble. And sorry, this allows it to be bound to tumor cells. They did use 5-fluorouracil as their therapeutic agent, and that's known to affect DNA synthesis. And so first they took these microbubbles or nanobubbles, sorry, they were quite small, and put them into a gel mold, and they were able to visualize them quite clearly using the ultrasound system. In vivo, they were able to take a look at the accumulation of these nanobubbles, and they used actually all 3 imaging modalities to ensure that the same type of increase of the microbubbles and retention of the microbubbles was seen across all 3 imaging modalities. So again, fluorescence, ultrasound, and MRI. Specifically with the ultrasound, they were able to visualize the accumulation of the nanobubbles over time and the retention as we see them out past 2 hours. And at this point, the blood pool level would have been almost back to normal, where we're obviously seeing the sustained retention of the nanobubbles, within the tumor. They then wanted to affect the release of the 5-fluorouracil to look at its effect on tumor growth. And so what they were able to do was create an acidic pH environment by hitting the nanobubbles with a laser. And by causing this irradiation, the acidic environment would actually cause the release of the 5-FU rather than the longer form of that agent. And so they did a number of different therapies, and they looked at tumors that had already been formed prior to entering the animal into the study. And it was only the nanobubbles with the laser which actually reduced the tumor volume. The other ones slowed the tumor volume, but they didn't actually reduce the tumor growth. They just simply slowed its growth compared to the control agents. And what they were able to do also is show that in using these agents, there was really no effect on body weight, which would indicate the animal's well-being. In this paper, the group was looking at hepatocellular carcinoma. And this is one of the most common malignant tumors, and it causes significant mortality. And so this group wanted to take a look at the synergistic effect of combining conventional chemotherapy with a gene therapy. And so hopefully, again, causing that synergistic effect. So in this case, they used oxaliplatin, which has an unknown mechanism, but it's believed to interfere with DNA synthesis pathways. They then also used a recombinant human adenovirus, ASPP2. The ASPP2 binds to p53, and it actually acts to inhibit cell growth and induces apoptosis. And they looked at the hepatocellular carcinoma cell line using a xenograft tumor model. And so here what they found was that using the combined therapy, that it caused the most significant decrease in tumor volume. All of the different treatments individually, or the 2 treatments individually, caused a decrease in cell growth, but it was most significant when they were combined together. This group also used power Doppler to look at the tumor vasculature. And although the data is not really shown here that clearly, they were able to find that the vessel quantity did decrease in all treatment groups, but was more significant with the combined treatment as well. just before we move to the next poll, I wanted to share with you some images that I took yesterday. Obviously, during the COVID pandemic, we're trying to find new and innovative ways to work with you guys to be able to see the systems in action when we're not necessarily able to travel or come into your labs. And so what I did was I took some blackberries from the grocery store, and I chose blackberries because they have a skin which we can penetrate with ultrasound. There's a fleshy part associated with each one of the balls on the blackberry, but they also have an inner seed, so it allows us to see internal structures and different textures. And what I did was I implanted a bit of the blackberry into a gelatin mold. And so this is simply clear gelatin placed into a mold from my children's science kit. And then I imaged them. And what I wanted to do with this was really perhaps provide an option for you to see the system in action, although not on an animal, but allow you to see how simple it is to use the system, interpret the images, and then find a way forward if that's of interest to you. And so this is an example of the B mode image that I was able to take. So the gelatin that they're embedded in is actually clear on ultrasound because it's very much a liquid material. We can then see the skin of the blackberry, the fleshy structure, and the internal seed is very hard, and it actually reflects all of the sound, kind of like a bone on ultrasound. And so we lose the signal below it. Then also did a three-dimensional image of this, and we can see within the cube that I was able to move the slices all around to look down from the top and see the different blackberry balls and coming at it from either perpendicular slice as well. So passing things back to Sam, one of the questions here will be if you're interested in a virtual demo, I'm happy to show you how this would work. And as we're able to hopefully get back into the labs here at a local lab, we can of course set up a virtual demo with animals when that is possible as well.
Yes, thank you, Tonya. And that poll is, which of the following would you like to learn more about? I apologize for the repeat on the first choice, but the choices are if you'd like to learn more about high-frequency ultrasound in cancer research, other imaging modalities available from Scintica that would include MR imaging, PET-CT, bioluminescence, confocal microscopy. Would you like a virtual or in-person demonstration of any of these imaging systems, all the above, or are you just here for educational purposes? We'll give 10 more seconds for others to answer. And I'd also like to encourage everyone to ask any questions at the Q&A dialog box below. We will address those questions at the end of this presentation in a few moments. I think mostly everyone has voted, so I'm going to end the poll and I'll bring back Tonya.
Great. Thanks again so much, Sam, for that, and also for you guys for answering the questions. So let's finish up with two more papers published using the Prospect T1. So this group again wanted to look at a targeted ultrasound contrast agent, and they wanted to combine the advantages of being able to visualize the tumor vasculature, but also be able to look at specific biomarkers. And that's really the expression of a specific marker on the surface of either the endothelium or the cell, the tumor cells themselves, with high sensitivity as well as specificity. And this group again used nanobubbles, and we'll talk a little bit about the difference between microbubbles and nanobubbles and why they're used. But specifically, this group functionalized it against two targets. The first one being vascular endothelial growth factor 2. And we know that that's expressed highly on the endothelium of newly formed blood vessels, so typically those that would be formed within newly developing tumors. The second is HER2, and so that's a specific target on the surface of breast cancer cells, as an example. And so moving back a little bit to the difference between microbubbles and nanobubbles. Traditionally, microbubbles are 2 to 3 microns in size, and they stay within the blood pool. They don't leak out into where the tumor cell matrix is. And where that becomes limiting is that the targets that you're able to detect or that you're able to bind to are limited to those that are expressed on the surface of the endothelial cells. Nanobubbles, however, are small enough that they can extravagate into the tumor microenvironment and actually bind to tumor cells themselves because they're small enough. So the VEGFR2 keeps us within the microvasculature. HER2 would allow it to bind specifically to tumor cells. And so what this group did, both in vivo and in vitro, was show that the dual targeted contrast agents bound more strongly in both situations. And so here what we can see on the top is the increased signal enhancement over time of the nanobubble when it has the dual targeted antibodies on it, and the retention of that over time within the tumor cells. This compared to either non-targeted or microbubbles which had only one of those targets. Here we're seeing the non-targeted bubble here with basically no retention of the contrast agent over time. But what they did see was that there was more binding and retention with the dual targeted compared to the single targeted agents. And then again, like I was mentioning, the sustained increase in grayscale intensity out to 6 minutes shows the retention of the nanobubbles within the tumor and not just passing through with a microvasculature. As we see with the non-targeted, by 6 minutes, the blood pool agent is the blood pool level is almost back to normal. They also incorporated a fluorescent dye within these nanobubbles, and they were able to confirm all of the ultrasound results with fluorescence imaging as well. And they also took a look at the histological results to show that all three of the modalities correlated well together. And the final paper that I'll take a look at today, again, was looking at a theranostic agent. So again, that's something that's both therapeutic and diagnostic. So we're getting imaging capabilities and drug delivery in the same device. This time, instead of microbubbles or nanobubbles, this team used multi-walled carbon nanotubes. And so they targeted these to prostate stem cell antigen to both target and detect the tumors, but also to allow those nanotubes to remain within the tumor to allow for the delivery of the therapeutic compound. And in this case, they used doxorubicin. So PSCA, as most people know, is really only expressed at a very low level within the normal prostate tissue, but is highly expressed in other forms of prostate cancer, as well as even more highly expressed in metastatic prostate tumors. This group, again, used a subcutaneous tumor model to do the characterization of their contrast agent. And so they had their targeted carbon nanotubes, and they accumulated well within the tumors. And so what we can see here on the top is the accumulation of the targeted nanotube over time. This was significantly more compared to untargeted or nanotubes which had been blocked with an antibody. So most important to note here is the change in the y-axis scale is significantly lower than that of the targeted nanotubes. And so then this group took a look at the therapeutic efficacy of the delivery of that doxorubicin to these animals. And they showed that they decreased the tumor volume, but what they also found was that they reduced the overall toxicity of doxorubicin to the host animal itself. And we all know that doxorubicin can have an effect on the animals. So the more targeted delivery using the nanotubes allows a higher therapeutic dose at the site of the tumor, but reduces the systemic amount of doxorubicin flowing through the animal, and therefore helping the animal to thrive better. So I hope what we've been able to show you today in the discussion has kind of led us kind of down a winding path. So we first started talking about the different types of tumor models, the pros and cons of them. We touched a little bit on all of the imaging modalities that we offer here at Scintica, but then focused more in on ultrasound imaging, how it works, what we're able to do with it. And then we reviewed some of the most recent publications showing how researchers have used it. A lot of those looking at contrast agent development, but of course using ultrasound to look at tumor volume growth and changes as well. And so on that note, I'd like to open it up for the Q&A, and I'm happy to answer any questions that you have. And as Sam said, please feel free to use the Q&A dialog box to answer any questions that you have, and we'd be happy to address them.
Thank you, Tonya, for that wonderful presentation. Yeah, as some questions are rolling in, we're going to get to some of the questions that were asked during the registration. So the first question that was asked is, in some transgenic tumor model, there are earlier changes prior to tumor formation. For example, dilation of the pancreatic ducts prior to tumor forming. Can this be seen using ultrasound, Tonya?
Yeah, so if you remember back to the slide where I was looking at surrounding tissue, the cysts forming within the ovary obviously weren't tumors. They were a buildup of fluid. And another example that I've seen is within that genetically engineered pancreas tumor model. Often we're seeing dilation of the pancreatic ducts prior to the formation of the solid tumor within them. And so the pancreas typically has a very homogeneous and characteristic appearance that is all different shades of gray. However, when the pancreatic ducts become inflamed and fluid begins to collect in them and they dilate, they turn completely black. And so it's immediately obvious of the early changes that are going on specifically in that model. And then when the tumors begin to form in there, you get a more gray appearance of the solid tumor beginning to form. Likewise, within other models, you can see changes earlier. If there's changes in the kidney, for example, in dilation or fluid accumulation in the medulla or between the medulla and the cortex, those types of changes are easily seen based on the difference in echogenicity or the brightness of the signal that we're seeing.
Oh, okay, interesting. And another question I was asked is, for those who are new to ultrasound, how long does it take for a new person to start using ultrasound imaging to detect tumors reliably?
Yeah, it's a great question. And my background when I started with high frequency ultrasound was not in imaging of any kind. And so where I always start when I'm training people is to recognize normal tissue structures. And so we start to look at the organ of interest that may be there. If it's a subcutaneous tumor, that is very easy to do because the skin has a very characteristic appearance, and then you see a bulge forming when the tumor begins to form. And so what we always say, we typically would come on site and do a 2-day training with you and get you comfortable with operating the system, identifying the organs that are of interest to you, and then identifying tumors in the models that you use yourself. And so at that point, at the end of the 2 days, people are generally very confident in identifying a tumor versus not. However, we do recommend that you practice and look at many different animals because they can look a little bit different each time over the course of a week or two. And then at that point, I would say that you're ready to start a study and that you're not going to become a better imager over the course of your study because you don't want your results to be affected by increasing your knowledge of how to do the imaging. So 2 days on-site training with someone to help you get started, and then practicing on your own for a week or two, and then ready to go for a study.
Oh, that sounds pretty easy. Like all other instruments, practice makes perfect.
Of course.
We have other questions that have been asked. Tonya, is there any that stand out to you?
We do. So I think one, and I kind of alluded to this, that there's 3 different probes with different frequencies, and what is the frequency range and what is the resolution of those probes? And so like I mentioned, there are 3 different probes. There's a 20 megahertz probe, a 40 megahertz probe, and a 50 megahertz probe. And when we talk about the frequency of the probes, we talk about their center operating frequency. So there's a broadband frequency, but that's really our center operating frequency. Frequency is important. What's more important is resolution. And so the 50 micron probe gets us down to 30 micron resolution. The 40 megahertz probe gets us down to about a 50 micron resolution, and the 20 megahertz probe is on the order of 70 micron resolution. So we would chat with you about your research, understand the structures that you want to image, because again, as frequency increases, resolution increases, but depth of penetration decreases. So if you're imaging rats, you're typically going to go with a 20 megahertz probe so you have deeper penetration to be able to see the structures of interest. However, if your rats are all using subcutaneous tumors, then you don't need the depth of penetration. So it really comes down to a conversation to make sure that we do configure the system to work appropriately for the models that you're working with. If your research focus changes and a different probe would be better, those can always be added on after the fact. So it's not that the system is locked with the probes that you purchase. So that's there. There was a really great question of what are the advantages of ultrasound compared to optical imaging, and it's something that here at Scintica we offer both. So it's an important difference between them. So, and I often see them not as competing imaging modalities, but really as complementary. So if you think of an orthotopically injected tumor or a metastatic tumor line where you're injecting cells, you don't really know where they're going. To scan with an ultrasound system and hunt and peck for where your tumor is located could be a little bit challenging. However, if you're able to use optical imaging where the cells express luciferase and you're able to see generally within the anatomical structure of where that tumor is taking hold, for example, if it's in the liver or if it's in the lower abdominal cavity, for example, you've got a place to start looking using ultrasound. So I see optical as being a guide in terms of that the tumor is taken hold, and where it might be located, and then using ultrasound to really accurately assess the tumor volume. And where the tumor volume comes into play is that the expression of luciferase is really important, as well as the delivery of the luciferin. So if your tumor is getting quite large and it has a necrotic core, or is experiencing hypoxia or a lack of blood delivery, then those things can be affected. So a necrotic core isn't going to express luciferase, and so your number of photons is going to decrease. So although your tumor volume may be increasing or staying the same, the number of photons may be decreasing. You can confirm that with ultrasound. So they really do play kind of hand in hand with one another.
Complementary as opposed to like competing systems. Yeah.
Exactly.
If anyone has questions, we do offer both. So
yeah.
You can reach out to us. Any other question that jumps out to you?
Yeah, I'm just looking through them to look at the questions that come through. So one of the questions was, can this ultrasound system be used for central nervous system imaging, so brain imaging? And if so, would a transcranial window need to be put in place for this? So ultrasound, when it hits bone, is fully reflected. And so there's different techniques that can be used to allow you to use ultrasound in the brain. And so opening up a transcranial window is absolutely one of them, and you can see the soft tissue of the brain very well. Bone thinning can be used, and at that point you would drop to a lower frequency because you get better penetration. So there are ways. It's not the easiest way to use the system, but it is possible with the system. Another question is looking back at those PDX tumor models, or the patient-derived tumor xenograft tumor models, and if avatar mice are used for breast cancer studies too. I haven't read anything about avatar mice being excluded from any specific type of tumor. I would think that they could be used for any tumor model, again, taking the patient tumor itself, implanting it in an immune deficient animal, allowing the patient to go on a conventional chemotherapy, but then using the avatar mice, which are those that have the that patient's specific cancer implanted in them, and using different therapeutic compounds to screen the mice for an effective treatment that then perhaps the patient would move to. I don't know of any cancer cell line or cancer that would be excluded from using that. So I think it should be absolutely possible with breast cancer tumor cells as well. So there's another question here, taking a look at detection of lymphomas by looking at the lymph nodes. And so a T-cell lymphoma could perhaps cause changes in the lymph node, but maybe only a third of them are palpable. And absolutely. So we saw that one image of the lymph node. That one looked quite normal in terms of there's quite a fluid pocket there, a little bit of grayscale intensity, but as they begin to dilate, pre-palpable. So the scale on that was in millimeters, and so that lymph node was probably 2 millimeters in size. I'd have to look back at the image and the scale. It's quite small. And so as those begin to dilate, you're able to measure either in 2D or 3D their size, and then track those changes as they begin to dilate before they would become palpable. And especially those that are deeper, perhaps along within the abdominal cavity, wanting to be able to measure those, because those are far less palpable than the ones, for example, in the fat pads, which are much more accessible.
We have time for a couple more questions if people want to ask. I have a question, Tonya.
Sure.
What are the largest sized animals that can possibly be used for this system?
Yeah, it's a good question, and it comes down to what we talked about closer to the beginning of as we increase the frequency of the sound, we decrease the penetration. And so it's not really the size of the animal itself. It comes down to how deep is the structure that you're looking at and what sits above it. So for example, I used ultrasound on non-human primates to look at the eye and looking specifically at the cornea. And so that's very superficial. So we can use the highest resolution imaging to do that. Could I look at the heart of the non-human primate? No. So it just depends on where the structure of interest is located within which animal. If we're looking at going through something very thick like pig skin, Although it may be more superficial, I'm going to use the lower frequency probe to get better penetration to go through it. So again, it's an in-depth conversation about what the application is and choosing the right frequency of sound.
That makes sense. Do you know if we have any resources we can get to one of the attendees on contrast enhanced ultrasound?
Yeah, I'd have to look for some specifically. It's an area that I've, in my previous life, worked quite extensively with, and with the system that we have here at Scintica as well. I've seen the images from the microbubble contrast agents. Lindsay, I'll get back to you directly about that, and perhaps you and I can have a conversation that can help guide me in finding some information for you.
Okay, sounds good. I think that's all the time we have for today, Tonya, and everyone who has attended. Thank you very much for this presentation. And to be respectful of everyone's time, we're going to wrap things up. As mentioned, we will answer any questions privately, any outstanding questions, and any person who wants to continue to ask questions, we'll try to get to them at a later time and send the transcript to everyone who's attended. I'd like to thank Tonya again for her interesting and incredibly informative presentation. And I hope that you guys, that everyone walked away with the things we touched upon, the different types of tumor models, ultrasound versus optical imaging in cancer research, image-guided injections of cancer cells, and the publications that Tonya overviewed. Please feel free to reach out to us directly should you have any questions about today's webinar. Also, please don't hesitate to direct any questions to us, and we'll make sure that we pass them along to Tonya. Again, thank you everyone for attending today's presentation. Goodbye.